Hey everyone, welcome back to another video by Camroom 5. In today's video, we're going to be doing some actual biology practical questions, which is biology O-level P4. It used to be paper 6, but now it has to be paper 4. Now moving on. In the last video, we did talk about the idea that question 1 involves mostly investigation performed, and then you have to answer question based on data completion. Table completion, you have to estimate values, average, you have to take their sum, you have to take their difference. It could involve you suggesting various apparatus, or you could talking about why a certain apparatus was more suitable. It obviously depends on the accuracy. So for example, if you are saying that we needed more accurate results and we needed more accurate volume, so you will talk about pipette and burette. And if you are concerned with more accuracy of a thermometer, then you'll talk about a digital thermometer, or you'll talk about a digital pH meter if you want to measure the pH. If you're concerned about the volume of a gas, you will talk about the volumetric syringe, and then you can also talk about other apparatus based on which quantity you're concerned with. In estimation of values, you have to take the difference and you have to follow the sequence. In case of a limiting factor like in photosynthesis that also came in your biology paper too, you have to keep the idea that sometimes a correlation is not maintained absolutely. Sometimes increasing one factor does not increase the other factor also. Now, moving on. Let's see this question. It says measure the length of each piece of potato in figure 1.1 and record your measurements in table 1.2. So I'm not doing it right now, but you have to keep a note of the fact that you will be given some diagrams of a potato piece, and then you'll have to measure that. Or it could be diagrams of apparatus, and then you'll have to measure what volume or what gaseous volume is there. So you can see that. So complete the last column in table 1.2 by recording the change in length. So if you put your final lengths here, you can easily find the change in length by subtraction. The part 3 says, construct a graph to show the effect of fruit juice concentration on change in length. So here, you can see a graph is already drawn on y-axis, change in length of potato pieces there. And on the x-axis, you will be putting the fruit concentration. So you will be putting fruit juice concentration in the units of percentage. So you can do that also. Moving on. Question 1 also involves improvements and suggestions on why errors were recorded. So I have done some data already for you, and let me show you how we can find errors and improvements. You can see over here that sources of errors are here. So let me show that this effort I did for you guys. So let's do it. The starting parameters are not similar in setups. Now what does it mean? If you're doing two experiments and if you want to compare them, you have to make sure that their starting point is similar. I have elaborated it by saying that maybe two experiments you began, but their volumes, their liquids, their solids, their specimen, they weren't at the same temperature in the beginning. What if you were analyzing two different liquids and their pHs weren't same? What if you were concerned about two liquid chemicals and their concentrations weren't same? Or you hadn't taken their exact same volumes? For example, if you said, okay, I am taking 10 centimeter cube, let's suppose you said I am taking 10 centimeter cube solution A in 2 test tubes, let's say. But what if you weren't exactly accurate? What if in test tube 1 you had taken 10.1 centimeter cube and in the other test tube you were at 10.3? It means you weren't as accurate as you thought you were. So you have to make sure that your starting parameters of both experiments or multiple experiments are the same, so that the comparison is more appropriate, and you can also use the word comparison is more fair. The specimen taken, whatever you are doing the experiment on, what if their identities and origins are not the same? So you have to talk about the idea that the specimen taken are not defined with their identity and origin. What if you are comparing two different plant species? Or what if you were comparing two different seeds? What if you're comparing two different fruits or two different animals? What if they were not comparable in a way that you wanted them to be? Let me give you an example. If you want to analyze example, to analyze the effect of temperature on seed germination, okay? And you said I am taking, let's suppose sunflower seeds. Let's suppose I'm taking sunflower seeds. And you said I'm taking them at 40 degrees Celsius, at 60 degrees Celsius, and at 90 degrees Celsius. What if your sunflower seeds are not exactly the same even in the beginning? What if at 40 degrees your sunflower seeds were fresh, at 60 degrees they were fresh, but at 90 degrees the sunflower seeds you used were stale? Or what if they were damaged? Or what if they were not even sunflower seeds at all? What if they were seeds from some other species? That means that in your experiment there's going to be an error because you claimed that you're using sunflower seeds, but you're not. So you have to keep a track of the identity and the origin of the specimen, like food source, like animal. If you want to compare, for example, the growth rate of two worms, you need to make sure that these worms are from the same kingdom, from the same species. For example, leaves, food source, food material, chemical specimen, they should belong to the same group, age, sex, and source. I'll give you more examples on that, by the way, don't worry. Another source of error is external conditions vary. For example, what if your temperature wasn't maintained constant throughout the experiment? What if the pH changed? What if the air pressure changed because in laboratory there was some window open? What if a fan was turned on? What if the fan was turned off when you did not want to turn it off? The wind velocity could bother the experiment. You need to make sure the concentrations are same and they don't change. What if they change? Surface area has to be maintained. So things like these should be maintained. Another source of error can include, in case of experiments requiring movement, when you're analyzing the movement of something, a very important error is the human eye, which is having a very longer reaction time. It has a longer reaction time. So when you want to see some movement, it has to be quick. And sometimes human error comes into play. So you have to talk about the idea that maybe the human error, maybe the reaction time was problematic. I remember in one experiment, it was a question about that a person is holding a rod and he drops the rod and another person grabs it. So another person has to grab the rod and someone was timing it. So someone was timing that how much time is required to catch the rod by the second person. What if the reaction time for the second person is really huge? So you can use video recording here and then you can, play it in slow-mo and then you can check how things are working. In case of human handling, human errors is very problematic. So talk about using more digital apparatus rather than putting everything on the human. Instead of asking the human to measure the temperature, to suggest that to monitor the temperature by their finger or by their elbow, use a thermometer. Instead of asking your colleague or your fellow to suggest the color of something, use a colorimeter. Colorimeter is also a very good apparatus to measure color intensity. In a similar way, use different digital apparatus rather than relying on the human senses. In case of a single experiment, you can see the last point, 6th point. Data handling is very important. So you can perform the experiment multiple times and then you can, take the average. So in case of single experiment, data handling is often incomplete. So more replicates should be there. You can connect the experiment multiple times and then you can take the average. This will, taking the average, increases reliability. Remember, reliability means You are more sure about the results because you have performed it multiple times. It does not increase the accuracy. It does not increase the accuracy, but at least it increases the reliability. So there are more questions, and we can continue to work on this document because I have made this document after much analysis of paper 6. We'll talk about it in the next video. Stay tuned, guys. Thanks.